In this video, I am going to discuss these problems and the limitations associated with Anfinsen's thermodynamic model of protein folding. So this is second part on the model of protein folding given by Anfinsen. Therefore, I will advise you for a better understanding of the topic that you watch my previous video on Anfinsen's protein folding model. Now, in addition, I would like to emphasize that from examination point of view, this topic could be good for a short notes and MCQs as well. So to identify the problems or limitations associated with his protein folding model, it is important that we review his propositions one by one. According to him, protein folding is guided by amino acid sequence or the primary structure of a protein. It does not require any accessory proteins or enzymes, and therefore the reaction is spontaneous. Second point is it is guided by thermodynamic parameters such as minimization of free energy change delta G and also maximization of the entropic changes of the system. It means that the folded or native conformation is the most stable form of a, a protein. We will review these one by one to identify whether these are relevant to in case of other proteins or enzymes available in our cell. So this is the scheme of his experimentation, in which he used RNAs in active, which is native conformation or folded conformation. When he denatured, it turned into non-native and inactive state. He was able to renature it successfully into the native conformation. It means that the two states are interconvertible. The non-native state is transformed into the active form or native conformation because A, it is guided by amino acid sequence, and B, the native conformation is the most stable state. The question arises is that this pathway does not operate in many other enzymes and protein. Once an enzyme is denatured, it fails to renature. It will not be transformed into native conformation. It means that it it contradicts or it challenges the hypothesis or the feature of his model that protein folding is guided by amino acid sequence. Second, that the folded or native conformation is the most stable. In these two conditions, in case of many other enzymes, the amino acid sequence remains the same in native and non-native. Then how come the protein or enzymes are trapped in a denatured form? This is not explained by the Anfinsen's thermodynamic model of protein folding. So this was the first limitations of his dogma. So the second limitation of his model is that it does not explain the role of co-translational modifications on protein folding. If you see his model, it is more toward post-translational protein folding. One step reaction, you have linear amino acid sequence or primary structure which is converted into 3D or native conformation. However, if you see the real world, a real cellular world, you will see that the protein is synthesized and modified simultaneously. When protein is being synthesized, they are also modified, and these modifications such as glycosylation and phosphorylation, they are required for a protein to acquire proper 3D structure or native conformation. However, the role of these modifications cannot be explained by his model. So this remains to be the second critical limitations of his model. So the next limitations come directly from a stark difference between Anfinsen and in vivo conditions where protein synthesis, co-translational modification and protein folding occurs. If you see his model system, it was very simple. It was very simple, having few chemicals including RNAs, beta mercaptoethanol, and urea. However, if you see the cellular environment where protein synthesis happens in cytoplasm as well as in the endoplasmic reticulum, cellular density is very high, and when protein is synthesized, it is not fully folded, it is not folded compactly, and therefore it is likely to interact with the cellular resident proteins. So how this high density protein impact the protein folding is not or cannot be explained by his model. In addition, it is now known that when a protein is being synthesized, tend to aggregate, interact with resident protein and tend to aggregate and denature. To avoid that from happening, cell has evolved specialized machinery consisting of chaperones, or these are the proteins or shaper molecules. What they do, they assist a protein to acquire, to fold, unfold, or to acquire proper 3D or native conformation. So the role of cellular protein density as well as chaperone cannot be explained by his model. In addition, effect of other denaturants such as pH, temperature, and ionic strength cannot be explained by this model. So, so in summary, the main point of his model is that protein folding is A, governed by amino acid sequence or primary structure of the protein, B, it has the least energy delta G values, and it is the most favorable and most stable conformation. However, protein folding, like his experiment, is not always post-translational folding. It involves co-translational modification. The many proteins undergo co-translational modifications such as glycosylation and hydroxylations or phosphorylation, which guide a protein to acquire native conformation, correct folded state. However, the impact of these modifications are not evident from his model. In the cell cytoplasm and the endoplasmic reticulum where protein synthesis happens, cellular protein density is very high. How this cellular protein, high density of cell proteins impact the protein folding cannot be explained by his model either. The effect of other parameters such as pH and ionic strength, which also influence the protein folding, cannot be explained by his experiment either. Now, when a protein, it is known when a protein is synthesized is loose, they are not compact, they are not folded, they undergo co-translational modification, then they become folded, right? So when a protein is not folded, is not compact, they may interact with the resident cellular protein, leading to their aggregation, precipitation, or denaturation. To overcome this, the cells have evolved special machinery involving chaperones which help the proteins to acquire native conformation. However, the effect of chaperones on protein folding cannot be explained by his experiment or by his model. The next point is when proteins, there are large number of proteins or many proteins, when they are denatured, they cannot be renatured, means they cannot be converted from non-native state into native state. While the protein sequence remains the same, why proteins fail to acquire native conformation cannot be explained by his model as well. And lastly, if you calculate the delta G value for a folded protein, it suggests that the folded protein is only marginally stable. It is not the most stable conformation. So these are these are the points which are so-called limitations or challenges to his model. I try to explain in the easiest way so that you understand it. In the coming days, I will be presenting uploading more videos on this protein structure and function, so that you understand at least this topic of protein folding and protein organization very well. So if you think it is helpful for you, please like it, subscribe it, and share with your friend who are interested in learning biochemistry of protein. Until then, take care and bye bye.